Great pleasure for today for me to to moderate the lecture that's going to be given by Dr. Achour. And as part of this particular introduction to the field, I'm going to have at the beginning 5-10 minutes to bring the angles that actually highlights the importance of what Dr. Achour is going to tell us. As Zoe mentioned, I am currently director of the Center for Applied Pharmacokinetic Research, but also I am Chief Scientific Officer for Certara and lead their R&D activities as well. Next, please. So today's presentation is in relation to individualization or personalization of the dose. And this is something that sometimes we do not actually consider. May I have the next slide, please? This is something that is a philosophical side. You look at many aspects of your life and you see that many elements of what you actually consume, you wear or whatever else, they are personalized for your taste, for many other attributes that you have got. Unfortunately, when it comes to medicines, this is not as much, let's say, paid attention to, even compared to the size of the shoes that you are having. Has got a better, I would say, individualization and personalization. Next, please. So this philosophical question is in the face of the human variability, and this variability is not something that is new. It is over 100 years that as part of the modern medicine that we have had statements that they are indicating that this variability is part of the life dealing with the patients, and the fact that the great physicians, that they don't look at the disease as just a disease, but they look at the patient and treat that patient who has got the disease, which indicates that in each case you may consider things slightly differently, and the therapeutic element with the drugs is not an exception. Next, please. The variability, as I said, it was recognized, but there have been a couple of, I would say, significant changes, as you can see from this graph that I have taken from my colleague Jeff Aronson of Oxford. And the cliff change that you are seeing in late 60s, early 70s were on the back of this particular book that came from Sir Mike Rawlins, who has been the head of NICE, National Institute of Clinical Excellence in the UK for several years. And this basically was starting to indicate what the elements of this variability in the therapeutic response to the drugs are. Next, please. And since then, there were lots of efforts with regard to the source of the variability that we are seeing. And these sources, they are not just genetic, even genetic, here in a funny version, this is coming from actually a picture in a book that's given there, and I have taken this slide from Jeff Tucker. As a funny version of the genetic importance, obviously is seen in the fourth guy from the left-hand side. But it's not, the genetic is not the only reason that people differ. The disease, the degree of the disease itself, environment and so on, the type of diet that we are eating, all of them, they are playing a role. And this brings me to the right-hand side of the graph that comes, this is a great, sort of reading from Xiumei Huang and colleagues in FDA, Bob as well, Bob Temple, that this is concern when you are developing the drugs, which is something that many of the members of ISSX, obviously, they are involved with. Next, please. And the sources of variability, when we look at them, you could roughly divide them on the variability in the PK side and variability on the PD side. We have made lots of progress on understanding the sources of the PK variability, but unfortunately, we haven't made similar level of progress with the PD side. This has caused some of the pharmacologists actually, on the back of considering PD as a bigger source, sometimes to ignore the level of the PK variability that we have to deal with. And if you want, to see some of the debates with regard to the PD variability from the PK and distinction between the local PK versus the PD that sometimes get confused, I recommend the exchange that we had with late Bob Forrest in CPT in 2013 as a follow-up to one of my papers. And these basically highlight something that those who listened to excellent lecture by Professor Josh Unatkat in AAPS earlier this year. The local PK and the role of transport is something that we are getting recognized more and more, and this is previously considered as variability mixed with the variability of the PD, while in fact it is PK variability and we can deal with. Next, please. So having said all of these, yes, the therapeutic effect, has got, many sources of variability, and a dose adjustment might be required. In many cases, we can, as a physician, you might be able to look at the outcome of the therapeutic effect of the compound and actually adjust, and that doesn't require anything, particularly if it is immediate. Sometimes, however, this is not possible, and then you have to go with the best known concentration that gives you that particular effect. And whenever this is related to the clearance, you can link it to the attributes that define the clearance. If it is renal clearance, it is relatively easy. Creatinine seen as a general function for the kidney, while when it comes to hepatic, we haven't got a general indicator that defines all different pathways, while in kidney even we have got some transport as well as glomerular filtration and so on. Most of them, they are at the moment considered that they are in parallel. Even we are progressing there and seeing that there are some discrepancies, particularly in ontogeny elements, etc. However, when it comes to liver, this is our major challenge. And next, please. And knowing that majority of the drugs in the market, in many different surveys that they are listed in the references, they are indicating that the CYPs and UGTs, they are playing a big role. It is important to identify the activity of these for each individual if we are really going to personalize the concentration for those drugs, knowing that different drugs are going to go through different pathways. Next, please. And for this, we have had many in the past, ISSX members, many will be familiar with the approaches that we have applied in the past. Of course, genotyping has always been there. We always have got, a number of different, seminars, workshops, etc. on that angle. But there are many of these enzymes that they are not under necessarily genotype control. And besides, the variation within each genotype could be also high. The phenotyping on the basis of individual probes or cocktails also is common, but it's not very practical. And many people have been now focusing on the biomarker as that they are coming as endogenous, therefore you don't have to give them separately. But this is again is not that specific in many cases, and also it's not showing the individual differences, but it is within individual. When they are interfering, they might, for instance, indicate that the induction is happening or suppression and so on. Next, please. So this is a typical thing that we will have with the genotyping versus phenotyping. Those bumps that you are seeing, they are the typical phenotypes that we call them poor metabolizers, extensive metabolizers. But within each of those groups, you have got genotypes that they have got within themselves lots of variations. Next, please. And this is what we want to identify. The cases for the biomarkers, endogenous biomarkers also have been put there, but again, they have been challenged in many cases. As I said, specificity is a problem, as well as their connection as a linear function has been questioned. You have to do some kind of modeling that makes them, more difficult, to interpret sometimes. Next, please. And the extension to the transporters are actually answering some other angles of those biomarkers, particularly with regard to the transporters in kidney that our group in Manchester has been working alongside many others, as well as liver side and so on. Next one, please. What we are trying to achieve is basically characterization of the individuals to a degree that we have got a better indication of what to expect from the admit process in that person. Whether we use this characterization in what's so-called, the PBPK analysis, which is the top-down consideration of the covariates, or next, please, or we consider them in the form of what I will later on, I will come back as a bottom up. The problem with the covariate analysis is that even the technically that is available there and many people can use it, the current medical records, they do not have necessarily the right characterization, that data that goes into these models. Therefore, you can see that in this analysis, when we looked at Dr. Darvish a few years ago actually looked at this with regard to the oncology area, majority of the models, the main things that they are considering, they are involving, let's say, body size and sex and age and so on, which they are, yes, covariates, but they are not the main determinant of an individual characterization of what is happening to their ADME, because we know they are related to the function of their enzymes and transporters and so on when it comes to clearance, as well as the first pass effect for absorption. The next one. So the idea with the PBPK is after the event. The only difference that the bottom up is having, it is still requiring the individualization and characterization. However, this characterization is actually from the first principle, from knowing the affinity of the drug to enzymes, transporters, that they are worked out in the in vitro systems, in organ-on-chip and so on. They are actually feeding from the bottom up and defining in advance that what we should be expecting in each individual. So the bottom line of what I have tried to actually to bring to you is that for individualization of dosing, we require characterization of that individual with respect to all the elements that they are influential in what is happening to the drug, how it comes into the body, how it circulates in the body, and how it gets out of the body. And so far, we haven't had many practical ways to actually roll them into clinic. Next, please. And with this background, I hope I have been able to highlight the importance of our lecture today with regard to the liquid biopsy, which enables quantification of abundance of the enzymes and transporters in human liver with relatively non-invasive. Of course, you need the plasma sample, but you are not punching into the liver. Today's lecturer, Dr. Brahim Achour, I have had the privilege of working with Brahim throughout his PhD, as well as the several years that he worked as a postdoc and now as a research fellow at University of Manchester. He has been very active in this area and in the translational side of the proteomics and various kind of omics. And it's a privilege to actually introduce you, Brahim. And without any further ado, I would like to invite you to take the floor and tell us this particular CPT paper, which is now out, is what's this all about. This was supposed to be presented in the Geneva ISSX European before publication, but now we have got the privilege of the paper being out, so people can actually go and read it after the lecture and understand further details. Over to you, Brahim.
Oh, thank you, Amin, and thank you to ISSX Education Committee for extending this opportunity to us, and also thank you to Satara for sponsoring this webinar. So to go into the crux of the matter, really, so Amin has highlighted that there are several ways that we have implemented in the past in relation to our attempt to individualize therapy. And these have their pros and cons, but most of them fall short when it comes to individualization. So we started thinking about novel ways that we could use to individualize patients. And obviously, we cannot use tissue biopsies for practical and ethical reasons quite routinely in the clinic. So the solution for us, or the idea that we thought about, was to use liquid biopsy as a way of individualization. So what is liquid biopsy all about? In simple terms, liquid biopsy is any biofluid that can be sampled from a patient in a clinical setting and can serve a clinical application, that whether it be therapeutic or a diagnostic application. I'm showing a few examples here of liquid biopsies that have been used before, and I'm showing the levels of invasiveness associated to them, from the least invasive to the most invasive. But you have to remember here that all of these biofluids are less invasive than tissue biopsies. The blood has been the most widely used liquid biopsy, and therefore I'll focus on that. And applications have only been focused on diagnostics, especially in the area of oncology, and I'll explain that in the next slide. So before we go into the sort of the technical aspects, and I'd like to explore the reason or the nature of a biofluid and how it can relate to tissue. So I'm showing in this cartoon here at the bottom the liver, and I'm showing a small section of the liver tissue, which is highly perfused in blood. And therefore, the liver is shedding small microvesicles, or what we call exosomes, that make it into the blood circulation. And the idea is to capture those exosomes that contain a snapshot of what is going on in the liver tissue, to capture those and to analyze the cargo that includes proteins and RNA, etc., that reflect the biomolecular state of the liver. And that was the challenge for us, really. So before I go into the science, I'd like to explore the market of the liquid biopsy. These are recent headlines from the news that were published over the last few months, September and possibly some in August, that show that the liquid biopsy technology is gaining a lot more traction than before. It is, as Amin would say, mushrooming everywhere. There is a lot more investment and I'm showing here only the North American market. There is more investment and more interest in this technology, and there are a lot more products that are making their way into the market. I'm showing a summary of those products here in this table that I have collated over the past couple of months. And I invite you to look at this table in your own time when the handouts are actually published by ISSX. But the take-home message from this table is really that although the liquid biopsy is gaining a lot more attention and attracting more interest, it is so far being focused on diagnostic or prognostic applications in the area of cancer, and therefore there is a gap in the market for therapeutic applications, for applications related to precision medicine, and to applications related to pharmacology. So to explain the biological principles behind how we developed our own liquid biopsy, I'd like to show you this cartoon, which summarizes how we sort of delineated the covariates related to expression in a liquid biopsy. So your enzymes and transporters are expressed in the tissue, and they are shedded into the bloodstream at different rates by different patients, where we can capture them and measure them in plasma. Now, if you have two patients, for example, and the two of them have similar levels of enzymes and transporters expressed in the liver, you would intuitively sort of predict that the levels in plasma would be the same. This is inspired by our data, but what we saw when we analyzed the data was that the levels were not were quite different. And the reverse scenario is also true. When you see that there is the same level in plasma, it is not necessarily the same level of expression in the liver. And that is because the variability in expression in the plasma has two components: variability of expression in the liver and variability in shedding. And in order to connect quantitatively between the liver and the plasma, we had to come up with a shedding factor to offset variability in plasma. And this was a combination of variability in expression in liver-specific markers in plasma. And I'm showing a list of those here. And I'm showing you also the algorithm of the shedding factor that we devised. Now, when we measured the shedding levels in cancer patients and also in healthy individuals, we noticed that the distribution of the shedding was population specific, was disease specific, and therefore there was higher variability in cancer patients than in healthy individuals. When we applied the shedding factor, we had to come up with criteria to select our liver-specific markers and targets as well. So the liver-specific markers, I'm showing a list of those here, and they had to have a high specificity score. I'm showing the liver specificity here as a fold difference. They had to be highly expressed in the liver. I'm showing here the levels of expression in liver tissue. Each plus sign represents an order of magnitude, and they had to also be consistently detected in plasma. And I'm showing here the consistency of detection in our cancer cohort. Similar criteria were applied to the targets. The targets had to be key proteins in the pharmacokinetics of drugs. They had to be predominantly expressed in the liver. I'm showing tissue specificity here. And they had to be consistently detected in plasma. And based on this characterization, we came up with a proof principle set of targets and markers that we measured using the liquid biopsy technology. So when we applied the correlations between the tissue levels that I'm showing here on the y-axis and the liquid biopsy levels that I'm showing here on the x-axis, the correlations were different before and after applying the shedding factor. You can see the before in red and the after in blue here. This is, well, we have to remember that these are samples that are coming from the same patients, so the plasma and the tissue are matched. And as you can see, once we applied the shedding factor, the variability was reduced and the correlations were improved, so there was less scatter and stronger correlations. Now, this was, what I'm showing here is a proof principle set of 3 examples here. But this was applied to 97 liver-specific or liver-enriched genes, and we noticed that the correlations were improved based on the normalization to the shedding. And therefore, for the rest of the talk, I'll show only liquid biopsy measurements that have been normalized to the shedding factor. So how do we apply this technology? And the aim really is to stratify patients based on their metabolic capacity or drug elimination capacity. And I'm showing here a cohort. This is a theoretical example, really. So I'm showing here a cohort of patients who have different metabolic capacities, from slow metabolizers to fast metabolizers, etc., etc. And you want to characterize them and stratify them in different strata, which you will administer a specific dose to each of them. So applications of this stratification would be possibly enrollment in clinical trials, where you characterize your patients before you administer the drugs to them, or in stratified dosing in the clinic. Now, you could argue that sometimes we can apply genotyping. However, genotyping can only apply to highly polymorphic enzymes, and the liquid biopsy can serve both highly polymorphic and non-polymorphic enzymes. Example here is CYP3A4. So, and there is also the example of so the pathways in which the drugs are metabolized and in which different processes happen in relation to ADME processes are complex, and most times you have multiple pathways involved in the disposition and elimination of a certain drug. And therefore, the liquid biopsy can serve that application because it can capture different genes and proteins at the same time. And to demonstrate this point, I'm showing a matrix here of correlation or a quantitative link between expression of different genes in the liquid biopsy as shown here on the x-axis, and in liver tissue as shown here on the y-axis. And as you can see, the correlations are quite good. Now, we applied the stratification process to determine the extremes in the expression of those enzymes and transporters, with the test being the liquid biopsy and the reference data being the tissue abundances. And as you can see here from the ROC analysis, any curve that's above the diagonal line represents a reliable prediction. And as you can see here, we could predict the extremes in expression quite nicely using the liquid biopsy. Some of them better than others, but good and reliable prediction across the board. Now, if we apply the numbers to the scenario that I've shown earlier, you could see that we could identify the fast metabolizers and the slow metabolizers at very high confidence. And then we applied the PBPK model for CYP3A4 substrates, alprazolam, midazolam, and ibrutinib at different rates of extraction ratios. And we compared between two different scenarios: uniform dosing, where you dose the drug to the patients at the same level, or stratified dosing based on the different strata, where you adjust the dose for the top group and the bottom group. And what we saw was that the variability in AUC here, which is a measure of exposure to the drug, was reduced by half. So you could see that there was a drastic improvement in the precision of dosing. So going forward, the liquid biopsy test has the advantage of being able to monitor the expression of more than 20,000 genes in the same assay. And out of these genes, we could identify more than 500 PK targets, which include enzymes and transporters. I'm showing correlations here between the liquid biopsy and the tissue for several of these targets, including additional CYPs, UGTs, CES1, and carboxylesterase, and also other targets that are not enzymes and transporters, including FcRn here, which is involved in the disposition of biologics. So Amin has highlighted the debate in relation to PK and PD variability. However, the question that is more interested to me would be whether we can actually predict or monitor PD variability using the liquid biopsy. And the quick answer is yes. We could identify more than 300 PD targets in the liquid biopsy, which have different roles in cancer, inflammation, immune response, cirrhosis, etc. And we could correlate them, some of them, to the tissue in the liver. So how about applying this individual characterization in modeling dosage regimen design? As Amin did earlier, I'll start with the POP-PK approach. And as Amin said, the covariates that have so far been used in the past, including weight and sex and renal function, that have been implemented for individualization, have little sort of influence on the metabolic capacity or the elimination capacity of the patients. And what we propose is that in the patient module here, you would introduce covariates that are measured in the liquid biopsy that reflect the pathways involved in the disposition or elimination of the drug. However, the problem with this approach is that it is clinical data driven, and therefore you will require measurement of the concentration of the drug a priori to application of the model, and therefore you cannot do this optimization before administering the drug to the patients. And therefore, we believe that the way forward is really connecting the liquid biopsy to PBPK and IVIV as a way forward to apply this in precision dosing. And what we mean here is PBPK beyond the typical view of describing the concentration time profiles based on physiological parameters, and going more into predicting variable kinetics of drugs in special populations based on information that gives you the average individual in each group. And the advantage of this approach is that it can address population needs where there are no clinical studies conducted, especially in the areas of pregnancy, children, or disease populations. Now I'm showing this graphic from a paper that was just published by our group, which looked at the regulatory submissions in the past few years in relation to the growth of the submissions over the years. And what you can see here is that along with DDIs, special populations and application of PBPK in special disease populations has witnessed the highest growth in the past years. And that basically highlights that we can use this approach in the same or a similar way, using the connection between the liquid biopsy for individualization and the PBPK for the modeling and the designing of the dosage regimens. So how does that work? What we propose is that the data that are related to individualization would be used for the creation of virtual twins or digital twins, as some people would like to refer to it. Which has so far only incorporated some genetic data and a lot of demographic data. And what we propose is that we pick up the elements from the liquid biopsy measurement that relate to the drug that we are interested in, that can go into the virtual twin building, and therefore you could predict the more accurately using parameters that are related to the individual's capacity for drug elimination. Now, this approach does not require measurement of drug concentration a priori to the prediction or the design of the dosage regimen, and therefore it can be applied before you administer the drug to the patients. And that can serve both in clinical trials where you want to know the dose of the drug that you want to administer to a certain stratum of patients, or in a clinical setting where you want to or you want to follow the approach of stratified dosing. Now, this all relates to the theoretical or roadmap to application in clinic or in drug development. However, what I'd like to highlight in the next few slides is what has already been achieved so far in relation to evidence that can support the use of liquid biopsy in clinical trials or in clinical practice. And this piece here that was published last year by Roland and Rodriguez shows a really nice sort of supporting evidence in relation to the use of liquid biopsy in a clinical trial. And what they did here was they measured midazolam clearance in these patients, and they measured also exosomal CYP3A4 abundance at the mRNA and the protein level. And as you can see, there is a good correlation between the clinical measurement and the exosomal or the liquid biopsy measurement. However, this only relates to only CYP3A4, and therefore it cannot be translated or scalable to other drugs. And it only focused on healthy individuals before and after induction, and therefore it doesn't relate to disease. We've shown some data earlier that there is differences between the disease populations, and there is no matched liver tissue to validate it. So there is also this study that was published only last month by a group in Sweden, which shows the utility of using tissue biopsies. So what they did was they developed a PBPK model that's informed by proteomic data on individual tissue biopsies, and they measured uptake clearance of rosuvastatin based on 4 different transporters. And they tested this in a good cohort, 54 patients. So the clinical data here in relation to the to the clearance were compared to the predicted data based on the liver biopsy PBPK approach. And what they concluded was that this approach was quite effective in predicting the clearance of rosuvastatin. So we see the predictions were in agreement with the clinical measurement, especially after correcting for the effect of OATP1B1 genotype on activity. So this basically demonstrates that a biopsy, a tissue biopsy, can be used for individualization. However, as we know, this is not practical and it's fraught with ethical difficulties, and therefore maybe a liquid biopsy could be used instead. And as we have shown earlier, there is a quantitative link that we have demonstrated between the liquid biopsy and the tissue biopsy. So we see this as evidence that supports our approach. So this brings us up to our own modeling, where we developed a PBPK model for 100 virtual individuals using SimSip, and the data that we inputted into the model included patient information that you can find in electronic health records, for example, including both demographic and clinical data, and also measurements from a liquid biopsy. So the dose adjustment was based on individual CYP3A4 content measured by a liquid biopsy assay. And as you can see here in this graphic, this represents individual dose ratios for each patient. Then we measured the AUC for the 3 drugs that I've discussed earlier using uniform dosing, stratified dosing, and individual dosing approaches. And the exposure, or the average exposure, for all 3 approaches was more or less the same. And this is a good thing because this gives us a measure of accuracy of dosing. So when we looked at the precision of the dosing, what we saw is that the variability in exposure was reduced based on the individual dosing approach by up to 2.5-fold, which is quite a drastic improvement in the precision of dosing these drugs in these patients. And again, this is still highlighting the importance of only one pathway, which leaves us with the question of whether this could work when there are multiple pathways involved in the disposition or the elimination of a drug. And this brings us to our ongoing work, which is in collaboration with Geneva University Hospitals, where the Optimat clinical trial is taking place. The cohort includes more than 300 participants, and data that we have collected so far include the phenotype, demographic information, and clinical information. And the patients have been phenotyped using the Geneva cocktail for several CYPs and PGP. I'm showing a few substrates here, and the approach that we applied for the phenotyping was a stratified approach. And the ongoing work now is we are doing the liquid biopsy assessment for a subset of these patients to see whether we can establish a connection between the liquid biopsy and the phenotypes. So all of these efforts highlight that there is progress in the area of individualization based on a liquid biopsy approach or a phenotyping approach in connection with the PBPK model. However, we believe this will not sort of achieve much unless the regulators are on board. Here I'm showing guidance from the FDA that encourages clinical trials to be more inclusive of diverse populations. And this basically shows the interest with the regulators in including different populations in clinical trials. And this will inevitably include patients from different disease populations, with different metabolic capacities, elimination capacities, renal function, hepatic function, and different age groups. And therefore, we will need an approach to characterize these patients or these strata and to dose them based on an individualization strategy or stratification strategy linked to a PBPK model. Going forward to the concept of precision dosing or individualized dosing, there was a workshop as well conducted by the FDA last year, which aimed to define the need and approach to deliver personalized drug dosing in the real world setting. So this basically highlights that this is not in isolation from the regulators. People on the regulatory side are aware of this, and they are encouraging the application of these technologies. So going forward into uncharted territories for PBPK and personalized dosing. So we have, Amin and others have published a few papers recently in relation to the trajectory of going forward in relation to personalized dosing. And what we think some of the uncharted territories that we really need to, or uncharted issues that we really need to resolve is ways of linking between the PBPK models and the liquid biopsy, electronic health records within the decision-making support systems in a clinical setting. The other challenges include formulation issues. How do you deliver the right dose of the drug in an oral formulation to the right patient? The example of this would be if you apply, for example, your liquid biopsy to a patient and using a PBPK model, you predict that this patient will require 83 milligrams of the drug, for example, and the drug is only formulated in tablets of 50 and 100 milligrams. How do you go about that? And we think the technology is already in place. It just requires application. So there is already technologies out there like macro pellets of the right dose of a drug that can be formulated into capsules using robotics, and you can deliver the right dose of a drug to the right patient in that way. Also issues in relation to delivering the medicine to the patient, and this is possibly only one of the only benefits of the lockdown on the back of the COVID pandemic, where several technology giants have started joining the effort to deliver drugs to the patient. So I'm highlighting here Amazon Pharmacy, which was launched late last year, and we think this might serve the process of delivering the individualized dosing to the patient. So I hope from this lecture I have at least made you think about the following points. We believe that personalized dosing will require a minimally invasive test to characterize each patient, which should not be specific for a single drug or a set of drugs. And we believe liquid biopsy can serve that purpose quite nicely. We believe that we have established a link between tissue expression in the liver and liquid biopsy in plasma for several PK targets, and there is ongoing work in relation to PED and disease targets as well. And we believe that models have demonstrated the utility of using a liquid biopsy PBPK strategy in adjusting the dose to achieve similar exposure in patients with variable hepatic elimination, or so-called precision dosing. And finally, we believe the infrastructure for implementation of personalized dosing is starting to take shape, but will take some time and effort from multiple stakeholders. And finally, I'd like to remind you of this. Maybe I've said this earlier, but it's worth mentioning again. Liquid biopsy technology is a reality, and we think that we should get ready for it. And this was the main purpose of this talk, really. So before I conclude, I'd like to acknowledge my colleagues at CAPKR and the University of Manchester, also the NHS for supplying the samples, Satara for funding the project so far, and for providing support with the modeling, Life Technologies at Thermo and Illumina for providing support with exosomal technology and RNA sequencing technology. And also special thanks to Professor James Rothman from Yale University for advising this project, and the Satara executive board members for being instrumental to the progress of this project. For the ongoing work, I'd like to acknowledge CAPKR colleagues and colleagues at university hospitals in Geneva, colleagues at Illumina, and also the CAPKR consortium members who have voted this project to continue, and also who have been funding the next steps of the project. And with that, I'd like to thank you all for your attention and to take your questions. Thank you.
Thank you very much, Brahim. You did a marvelous job also keeping with time. We have got several questions, so I will be starting. I'm not sure people want their name to be mentioned or not, but I'm guessing that because their name is shown in the Q&A section, so they are happy for me to mention that. So Kimo Tohiyama is asking, what is the key in controlling the level of the shedding? Can you actually explain if you know anything about, what controls the shedding, the fact that it's higher in cancer, but, lower in, healthy, what else do you know? Or do you know anything?
So this is one of the areas that we've been asked about before, and really the differences in the shedding factor were not expected. We didn't think that we thought there would be variability, but we didn't think that the distribution would be population specific or disease specific. So the disease is certainly one of the factors that influences the differences in shedding, and we believe also some demographics like age and it could be a factor, but this is something that we might want to explore in the future, but it hasn't we do not really know exactly what's going on, and it might be a question for future investigations.
Thank you. Hopefully, that was adequate answer. And Nancy Hakuz is asking, can you explain that the exact nature of the liquid biopsy sample? Was it whole blood, plasma? What was the size of it, and so on? Did you have to have a 1 liter, sort of to do, all of this, what you have done, or, sort of, I don't know, 5 milliliter or whatever?
So what we think is the key factor here is extracting the exosomes, which are small microvesicles within plasma. So we would normally fractionate the blood. So we get the blood just before the surgery or fresh blood, and you fractionate it and you take the exosomes out of it. Now, you could use, well, some other technologies use the buffy coat or the cellular part of the blood. But we believe the best way of describing enzymes and transporters from remote organs would be the exosomes that are floating and coming from these organs. Now, in terms of the volume, I would say you could go down to 1 milliliter of plasma, or even lower than that, to extract sufficient amount with good quality, sufficient amount of RNA or protein with good quality for downstream application.
Thanks. Marcel Hopp is asking if there are any confounding element coming into any of your assays with regard to what is coming from intestine. Can you comment on that?
Yeah, so the understanding the biology when it comes to the liquid biopsy in connection with the modeling is really important. So the liver sheds into the blood. However, intestinal tissue would shed mostly into the feces, and therefore you would expect some contribution, but not a major one. So the contribution is quite low. If you're going, for example, with the kidney, then you would probably want to go with urine and blood or something like that. But we believe the blood is a good description of what happens in the liver.
There is also an anonymized question that is in the same line, and it is basically saying how you distinguish between your different sources of whatever that you are seeing in plasma. So how do you know that all the CYP3A4 is coming, or PGP is coming from liver versus, I don't know, any other, forget about the intestine, but, other organs, let's say?
So fishing out particular exosomes that are coming from different tissues has been tried before, but the literature that I have read so far is not very convincing. And therefore, we went with the criteria that I've explained earlier that makes sure that most of what we see in the plasma or these exosomes is coming from the organ, the target organ that we're interested in. And therefore, you could apply modeling to distinguish between the sources of variability. However, I would say at the moment we are not at that level where we could distinguish from between the different components yet, and therefore we're only sticking to the elements that we know are specific to the organ that we're interested in.
Brahim, you have got a special question from President of ISSX and Daily, and is asking if you have got any information on different shedding factors from different diseases of liver.
We are thinking thinking of investigating, and I see she actually mentioned a non-alcoholic fatty liver disease, and yes, we're interested in that, but at the moment we're in the first steps in acquiring those samples. The issue with this sort of technology is that to validate it, you really need the right samples, and therefore we are really interested in this, and we believe that there will be population-specific distribution for those disease areas. However, we haven't explored that yet.
The other question from Andrew Parkinson is the same line as the question from Marcel, and is basically saying who's going to take care of the samples in feces. It's not in that line, but it is basically saying, you know, SIPs and transporters in intestine, how we are going to actually do something about them, in your view. Some poor PhD student, not you.
Well, if it's exclusively expressed, so if the target protein is expressed in the liver and the intestine, then we have a problem. We have to account for the feces, etc., etc. But if it's exclusively expressed in the intestine, then you might be able to apply scaling factor from the elements that are shedded into the blood. So that's one way of going about it. Otherwise, you would want to have a desperate sort of PhD student do the feces-related work.
There's a question on the housekeeping proteins and their role in the shedding factors. I'm guessing that, you know, you actually can refer to the paper with regard to what actually consists of the shedding. But the shedding could be defined based on different targets. You worked on liver, I'm guessing, but you could have done it for brain or whatever else.
We've devised a theoretical framework for different organs, including the lungs, the kidney, the brain, etc. And we believe the housekeeping proteins have to be vetted quite extensively before they can be used to correct for the variability in shedding. That is actually right.
And the next question from Tang Wang, what biomarkers in the exosomes exactly you looked at? I'm not clear on that question.
I'm not clear. Yeah, exactly. Yeah. So we looked for we looked for, so on the PK side, we looked for enzymes, transporters, receptors that control the biologics, for example. So we looked at those and we characterized those along with the markers of shedding. And for the PD side, we looked for disease markers, cancer-related markers, inflammation-related markers, and immune markers. So this is in a nutshell what we looked for.
Janne Bachmann congratulates you and also is asking different CYP3A substance, how, you know, sort of you will build this PBPK towards different drugs, I am guessing. I'm guessing you can actually combine even not 3A4, you know, a little bit of 3A4, a little bit of 2B6, whatever the path we know for the drug on the basis of their affinity to enzymes, you can build that. That was my understanding.
It's not as simple as one drug being controlled by one enzyme in the clinical setting. So we believe that the power of the liquid biopsy test is that it can capture different pathways at the same time. And therefore, if we know the contribution of each enzyme or transporter to the disposition or the elimination of a particular drug, we can actually devise a more complex model for it. And we haven't done that yet, but it is part of the next steps that we are exploring.
But the combination are possible because you have got the data, you know, I'm guessing. Steve Leader is asking how actually you will be looking into the longitudinal effect. You know, what you have measured is obviously in a single time point where you had the biopsies of the liver as well as the plasma. But have you got any plans to look at, you know, whether you need to repeat, you know, the same measurement in the same person every 6 weeks, 6 months? You know, what is your idea in that line?
When we get to the clinical application, that will be required. That element will be required in the clinical application. Where, for example, if you're having a support system for physicians to to give a more precise dosing, you'll have to have an idea of the longitudinal or the temporal changes related to the liquid biopsy measurement. But we haven't done that yet. It was one of the plans that we thought that we would do, but we haven't had the time or the funding to do it. Is there any more questions, Amin?
Sorry, I was asking, you know, sort of, can you actually say whether going forward with the studies that you are planning, you will be doing this? Because my understanding is regulatory agencies also interested to know how regularly you need to do that. Every month, you know, every 6 months, how much it is changing.
Well, you'll have to understand the biology behind it, or you have to test it at least first. It might be every month, or it might be every 6 months. We do not know yet. I know that there are circadian changes in the expression of enzymes and transporters, etc., and I'm not sure whether that will be an issue later on for changes over time. But I believe that you might need to at least do this like 6 months or each 6 months to establish that the baseline stays the same.
Next question from Andrew Parkinson again. He's asking whether you can distinguish between a CYP that is basically reflection of the active enzyme in liver versus a CYP that is already, because of covalent binding or whatever, is actually completely, you know, knocked and is not active in liver. Can you do that distinction?
We do not do activity in the liquid biopsy test, but I believe if a particular enzyme is suppressed or induced, then you can actually measure the changes in expression. However, in relation to activity, you might want to do a phenotyping experiment for that.
So the inhibition is out of question, but if it is levels, yes.
Yeah. Okay. This is why we're trying to avoid inhibition in the cocktail study here.
The next question is in the same line. Marcel Hupp is asking whether the shedding is constant over time. So yes, what you will get from, I guess, Steve Leader's question is that the variability in plasma, but then if you want to answer whether it is coming because of shedding or because of the variable actually level in liver, then you need matching sample again.
Exactly.
You can't get that only based on plasma. That's
exactly.
my understanding.
And the variability might be related to the expression or related to the shedding, and you have to distinguish between the two, and therefore you would probably want to have a dedicated study where you get matched tissue with the plasma at different time points. As far as I'm aware, this has not been done before, so yeah.
I can't read the end, but of part of Andrew's question. Say what is about Manchester that is so attractive to what?
He's saying geniuses, but I'm not sure whether he's referring to you, Amin, or.
I am guessing it is, it is to you, Brahim. I have never considered myself in that category. Thanks, by the way, Andrew. Next question from Dr. Liu is how you done something like day-to-day variability? I think you answered that.
This is in relation to the biomarkers of shedding, for example. We went we selected stable sort of expression in the liver. So markers that are expressed at more or less the same level at baseline over time were selected to construct the shedding factor based on.
Anonymous attendee is asking why you have to do plasma sampling. Why not just do therapeutic drug monitoring? I think, can you go back to the prospective versus retrospective argument that you had, and the fact that this is not specific to a single drug?
Yeah, well, this is the here. Is it? Are we talking about this?
It doesn't matter. Yeah, I think they want to distinguish between therapeutic monitoring and measurement of the drug. For if you want to do for 100 drug, maybe I take this. If you want to do 100 drug in a hospital, then you have to have 100 assays in that hospital. And no hospital will have 100 separate HPLC running in parallel to measure them. Even if you had, 20, you have to, come up with a management plan. While in this one, this is not specific to drug. You get, the characterization and you apply it on the basis of what you know of the drugs to any drug, as long as you know the role of that particular transporter and enzyme and so on. So I think I could even
that is a translation, it's a translatability element to it.
Yes,
and also you have to measure the drug concentration beforehand to be able to optimize later on for TDM. So
I think this is another anonymous. It's a very good question. I have asked this before as well from you. Is there a known half-life for the exosomes? And of course this goes back to the fact that how they correlate from plasma to the tissue, while people even within tissue, they have sometimes failed to actually come up with the correlations. What's your answer to that? I believe that is in the discussion of the paper, but it would be good if you explain briefly.
Yeah, so what we believe is the exosomes tend to accumulate in the pool of the plasma, and therefore we measuring a time averaged or steady state level of these exosomes. Whereas the cargo of the exosomes really, but whereas in a liver tissue, what you see is the snapshot of the expression at a particular time point. So that's why we think the correlations were a lot better. In relation to the half-life of the exosomes themselves, it will depend on various conditions. So this will depend on the disease, will depend on physiological conditions, will depend also on the drugs that the patients are on. So we do not have a handle on this. But the expression itself, we are measuring the steady state level, time averaged level in the pool of the plasma.
Okay, so one of them is basically a damping effect on the, the, let's say, very high surges of the RNA that's happening in tissue. So in the tissue, it goes up and down much faster, while, it comes to the plasma and it's basically floating at a slower level. So a damping effect. Next question from Shinji Yamazaki, who's asking about, again, induction and TDI. I think you answered this with regard to the inhibition that I think Andrew Parkinson was asking, particularly time dependent and covalent binding. This cannot answer that, but with regard to the induction, as Andrew Rowland and Dave Rodriguez have already shown for the CYP3A4, that is actually answering. But with regard to the time element, I don't think you have got the data yet, and same goes with the variability to answer that. So if you just confirm.
Yeah, the induction case was quite nicely demonstrated by Rowland and Rodriguez in their paper. So that supports but the time delay, etc. We do not know. We haven't explored that.
Okay, the next anonymous question again is coming with regard to the isolation. What was the exact isolation methodology you used for exosomes? Did you use KIT or anything like that?
So there are different methods used in practice for isolation of exosomes, but the method that we used was a combination of polymer-assisted precipitation and centrifugation. So that there are other methods used in the literature, but we think this was more suited to the exosomes we're interested in.
Okay, Sagnik Chatterjee is asking, will liquid biopsy will be able to predict internalization of therapeutics as observed in the case of NASH?
The short answer is we do not know, but if it affects the shedding of the transporters into the plasma, then it might be able to.
And again, another question from Sagnik Chatterjee on liquid biopsy and whether the transporters, I think you actually, this is talking about canalicular transporters. Yeah, okay. I guess that you had them in some of your data. Actually, you showed them.
Oh, I've shown PGP, BCRP.
Yeah.
Yes, in the correlations between the liquid biopsy and the liver. But if we're talking about intestinal, then again we go back to was it Marcel Hopp or Andrew Parkinson's question? It will depend on the target, really.
Okay, I think we answered all the questions,
Stability,
and we have
one left.
one left. Okay, oh, there one new one again from Nancy Achour, I think. How stable these exosomes are after isolation? For how long?
So exosomes are quite stable after isolation. The problem is the cargo. So if you do not open up the exosomes, then they can be very stable for long-term storage, and also they can be very stable to freezing and thawing. So this is what we've learned from doing the work over and over again.
There is a comment from Andrew with regard to Andrew Rowland and Dave Rodriguez, and that is activity that they measured on the exosomes. I think, you know, they were measuring actual protein while you were measuring the RNA. So there is a difference here. So can you comment on that?
We have samples that we've isolated for proteomic analysis of the.
But what you were showing were not the protein. They were
no,
RNA.
this is RNA. This is RNA because it allows us to explore a wider number of targets at the same time.
So with that, we are now over time. In fact, you know, 7 minutes. Thank you very much for all those people who contributed. Back to Zoe.
Great, thank you so much, Brahim and Amin. So I just want to remind everyone to please complete your evaluation that you will see on your screen after the conclusion of today's webinar. Thank you so much. Take care.